You may proceed, Mr. Proactor.
Welcome, everybody, to this online defense. Hereby, I open this academic ceremony in which Christian Schmitz will defend the academic thesis, development of a fully enzymatic conversion process from marine chitin to chitosan oligomers. May I invite you to present a summary of your study and the conclusions of your thesis?
Yes. I will share my screen. I assume that everybody is able to see the presentation now?
Yes, we can.
All right. So good afternoon, everybody, and welcome to the presentation for the defense of my thesis titled Development of a Fully Enzymatic Conversion Process for Marine Chitin to Chitosan Oligomers. At the moment, the annual global shrimp market looks at a quite impressive turnover with approximately 20 million tons. However, 50% of that material amounts to the non-edible shell material, of which currently about 90% is just deposited in landfills or dumped in the sea or just burned. And merely 10% are biorefined to products of higher value. For instance, the proteins from the shell material are utilized as fertilizers or animal feeds, the calcium carbonate as products from pharmaceutical, paper, and textile industry. And lastly, there's the polysaccharide chitin, which is mainly used for the production of chitosan. Chitosan and its shorter chitosan oligomers are functional molecules with versatile potential and actual applications, especially in technical, agricultural, biomedical, food, and cosmetic industries, due to their very diverse antibacterial, antifungal, antioxidant, anti-inflammatory properties. These properties, however, are heavily dependent on the degree of polymerization and degree of acetylation of chitosan and chitosan oligomers. Typically, these molecules are produced from chitin by a chemical depolymerization and chemical deacetylation. However, there are multiple issues associated with that process, and it is very difficult to control it, especially considering the degree of polymerization and degree of acetylation, and as a consequence, the associated properties. And there is a high product contamination risk posed by residual chemicals as well as side products. One solution to overcome these issues would be the development of a fully enzymatic process where the chemical depolymerization and deacetylation steps are replaced by more selective and specific enzymes. This would give more control on the degradation process to achieve more consistent products, while simultaneously also reducing the risks for potential contaminations. However, such a fully enzymatic conversion process from chitin to chitosan oligomers has not yet been established yet successfully, and it's therefore the aim of this thesis. So for the development of a fully enzymatic production for chitosan oligomers, first, a suitable bacterial origin has to be identified that's capable to effectively degrade chitin. Further on, the responsible enzymes for that chitin degradation have to be identified, respectively chitinases and chitin deacetylases. Those enzymes will then be produced individually by a recombinant approach, followed up by a full characterization of the enzymes regarding their reaction optima as well as the product properties. The characterized enzymes will then be implemented for the development of a fully enzymatic process, coupled here with reaction optimization by statistical analysis. So the novel bacterium G5 was isolated selectively from seawater samples in Ostende, and it has a quite interesting growth characteristic as it can utilize crystalline shrimp shells, or respectively the chitin in the shrimp shells, as a sole carbonate nitrogen source. Further assessment of the strain revealed that it produces various chitin-degrading enzymes, namely chitinases and chitin deacetylases, and those carry out an extracellular degradation of the shrimp shells to small monomeric and dimeric compounds that are assimilated by the organism for metabolism. And as those enzymes from G5 are working very effectively on crystalline chitin, the strain was selected as the origin for the target enzymes for the novel process. First, a genome sequencing of the strain was carried out, and it allowed us to identify 5 genes that are coding for chitinases and 5 genes coding for chitin deacetylases, and those were cloned subsequently into Escherichia coli. Therefore, multiple gene constructs or enzyme variants for each of the individual enzymes were tested for their production levels and enzyme activity. And based on that, the best variants were selected for an upscale production for the chitinases, as well as for the chitin deacetylases. These optimum enzyme variants were then produced at the 1 liter scale in E. coli BL21 in shake flasks, and the target enzymes were purified and enriched successfully from the culture supernatant using affinity chromatography. The characterized chitinases were then, the purified chitinases were then characterized extensively using shrimp chitin powder as a substrate, and we were able to successfully identify temperature, pH, and salt optima, as well as temperature stability and the influence of multiple cofactors, so metal ions and detergents, if they act as reaction activators or inhibitors. Recording of enzyme kinetics revealed the substrate affinity of the enzymes and revealed also enzyme saturation effects. Product properties were determined after enzymatic hydrolysis of various chitin substrate, and it revealed that not all of the 5 chitinases were equally active on insoluble chitin as a substrate, and mainly dimeric, trimeric, and tetrameric compounds could effectively be generated. Similarly, the chitin deacetylases were characterized for the same parameters as the chitinases, and also here, temperature optima, pH, and salt optima were successfully identified, as well as temperature stability and the influence of multiple cofactors. Enzyme kinetics revealed that not all of the 5 chitinases were working effectively on crystalline chitin, and therefore also soluble chitin trimers were selected as a substrate to identify the properties. Analysis of degradation products after enzymatic deacetylation revealed that 3 of the 5 chitinases were effectively mono and deacetylating chitin oligomers. The characterized chitinases were then used for the development of a depolymerization process, and therefore multiple chitinase cocktails were developed to achieve, first of all, different product properties concerning the degree of polymerization and also to maximize conversion rates. Therefore, 48 different chitinase mixtures were modeled statistically and then tested in hydrolysis experiments using chitin powder as a substrate. Analysis of the product properties of these hydrolysis experiments revealed that independent on the enzyme mixtures, the polydispersity of the enzyme of the product mixtures could not be changed effectively. However, activity data of these experiments could be used to evaluate our statistical models, which ultimately allowed us to develop optimized chitinase mixtures, which resulted in an 80% increase of the conversion rates. And with this 80% of increase of conversion rates, we were able to achieve product yields similar to what is currently reported for chemical processes with approximately 30%. The products from these optimized chitinase mixtures were then implemented as substrates for the development of a deacetylation process. Therefore, again, multiple chitin deacetylase cocktails were developed to achieve altered degrees of deacetylation and also to maximize the conversion rates. Again, 48 chitin deacetylase mixtures were modeled statistically and then tested in hydrolysis experiments using the respective substrates. We were able to identify by the product analysis that a monodeacetylation of the substrate could effectively be generated. However, again, as similar to the chitinase mixture design, the product properties regarding the degree of deacetylation could not be changed effectively. Nevertheless, the activity data was utilized to evaluate our models, and optimized chitin deacetylase mixtures were successfully developed. This allowed, in the end, a 20% increase of the conversion rates, and ultimately, with the whole process at 30% yield, we were able to generate 95% monodeacetylated dimers, trimers, and tetramers from chitin powder. So to sum everything up, the new bacterial strain G5 was fully characterized and identified as a chitinolytic bacterium. Novel enzymes for the degradation and deacetylation of chitin were discovered within its genome, cloned and successfully expressed by E. coli. All enzymes were fully characterized on insoluble chitin powder, respectively chitin trimers for the chitin deacetylases, and optimized enzyme cocktails were developed for the production of monodeacetylated chitosan oligomers from chitin powder. Yields for these chitosan oligomers from the fully enzymatic process are currently on par with what is reported for the chemical processes. So that's all from my side at this moment. I want to thank all of the listed people here for their continuous support during the past 5 years during my PhD. And of course, I want to thank all of you for your attention. Thank you very much.
Thank you very much for your quick presentation. The opposition now will be opened by Professor Hacking. Professor Hacking is chair of the assessment committee, and he is also a professor at the Karen School for Cardiovascular Diseases here at Maastricht University.
Well, thank you, Mr. Prorector. So what shall we do with the extra 6 minutes? Shall I take them or shall I among the other? committee
They are
member.
all yours.
We'll see how far we can get. Well, dear candidate, first I would like to compliment you with this impressive thesis that actually feels like an exciting boys' book. Explore the North Sea at dawn, take a sample of fresh seawater in an Erlenmeyer flask and bring it to the lab, and discover a new bacterial strain and decipher its enzymatic pattern and then save humankind with it. On top of that, it's covered in secrecy as all is patent pending, and above all, it's a pleasure to read. I've learned a lot, and in all, your achievements actually make me a bit jealous. Of course, many of these compliments are also addressed to your promotion team. Today, I would like to try to challenge you a little bit on your depolymerization part of your thesis. And when I look at page 40 at paragraph 2.4, I understand that there are many bacterial strains that actually can degrade chitin, but that you actually say that novel strains of bacteria are essential to yield large-scale chitin oligosaccharides, especially the double ones, in contrast to these useless monomers. But when I look at page 60, in the conclusion section of your discovery chapter, you state that the whole strain cannot be used for the production of chitin oligosaccharides from marine chitin, as it generates mainly DP1 products. And this is also very visible in figure 3 on page 59. So your recommendation is then to unravel the different enzymes and to produce them separately, recombinantly, to see which one can be of use to produce these costs. And so my question first is, so your new strain might not be actually that different from the previous or older strain. So why not use one of the previous strains to do that? So why actually do you need this novel strain?
Highly esteemed opponent, thanks a lot for your question. I'm really grateful to answer that. Actually, to answer that in a quick mode, we didn't know. We didn't know exactly in the beginning what to expect from the strain because it was completely novel. And we just noticed that it can degrade effectively crystalline chitin, which is really an attribute that is of utmost importance, especially considering that lots of these chitinases don't work effectively on crystalline chitin. Okay. And we didn't actually know exactly what to expect. And that's also what this whole project is based on, to identify what's actually capable with these novel chitinases. Of course, the chitinolytic strain it's nothing special per se, because these are very essential in the carbon cycle in the marine environments. But nevertheless, especially considering that there are such a broad mixtures available or broad specimen available from the chitinases, and the fact that there is at the moment not yet an efficient process established just using chitinases to degrade chitin into oligosaccharides, I think we are really urged to use any available source of bacteria that we can find that looks interesting from this perspective, and to eventually utilize the information that we can gain from the strain to develop a new process.
Right. So, but the main advantage, as you already stated, is that this strain can use crystalline chitin as a source of substrate.
Yeah, it's actually that's of course an added value, of course, also to develop eventually even more processes around that. That's something I discussed also in the outlook of the thesis. But there are also, for instance, multiple strains of bacteria, also fungal origin, that also produces broad mixtures of chitinases, right, but they don't utilize them as a carbon source. So they're just degraded. It's not entirely known what's the exact application for that, or why the bacterium is doing that in the first place. Eventually as a defense mechanism, but the utilization of that as a carbon source, of course, is from our perspective, of course, an added value.
Okay. Well, this is actually convincing. So in the same chapter on page 61, this is just out of curiosity, you reveal the source of your new bacterial strain. Eh? So actually, was it actually you that sampled the water, and why in Ostend? So were you there on vacation, or so why was that?
Highly esteemed opponent, thanks a lot for your question. No, it was actually not me. It was isolated approximately 10 years ago by one colleague at the Fraunhofer IME. And in fact, but I know that this person who isolated the strain was just there on holiday, and she selected a random sample there on the beach. And eventually it contained such an interesting bacterium. So really interesting to see what the marine environments have in place for us by just randomly sampling it. Okay, yeah, that's really interesting. So, I mean, more strains could be available, like also like in the EC or wherever in the Pacific. So this is like all, yeah. Okay. Yeah, And that's actually also referring to one of the propositions that I have added there, that the marine environments are, to put it simple, an inexhaustible source to discover novel bacteria eventually. So also extremophilic bacteria especially. So lots of psychrophilic enzymes isolated from sea that are especially effective at low temperature, which is interesting, for instance, for food applications. But also barophilic enzymes or strains or hyperthermophilic bacteria are also found from deep sea samplings. I think also one enzyme that has the highest efficacy at 140 degrees was discovered actually from deep sea samples. And that is also interesting, especially considering industrial applications that have to be carried out at high temperatures.
Yeah, definitely. Okay, that's really cool. So then I would like to continue on the selection of your 5 enzymes, your 5 chitinases in your chapter 3. And like the first question is, how can you explain that your whole strain is able to generate DP1 products while your 5 selected enzymes actually are not? So they mainly generate DP2 products, maybe a bit of DP3. So why do you think that is? So have you been too selective, and actually did you create sort of 5 of almost the same enzymes? So what do you think?
Highly esteemed opponent, thanks a lot for your question. Yeah, that's actually also a very interesting question to answer, because it looks very surprising that the enzymes per se are different regarding their domains and also their potential structure, but still they resulted in similar products. And especially for chitinase 5, this was this chitobiase domain, which is actually known to hydrolyze dimeric compounds into monomeric compounds. It's something we would have expected to see there, but apparently this chitobiase didn't work effectively on the substrate. But the answer for this question lies not in the chitinases, but it most probably lies in the chitin deacetylases, because there was one of the chitin deacetylases contains a domain of unknown function. It was deacetylase 3. And as depicted in the thesis, apparently this domain contains a chitinase activity. So this poses the question whether there are even more chitinases within the genome of G5 that weren't even identified by the protein family database search that we did. So it's non-homologous chitinases, basically.
Right.
And eventually there might be one of those that we didn't identify yet that is essential for production of the monomers, because we saw some monomer generation especially during the chitinase optimization of the DOE experiment. But those can be considered as side products from the degradation of trimers. So it's not actively creating these monomers. Mm-hmm. They're considered just a cleavage product from the trimeric hydrolysis of those. But the actual enzyme, the exoenzyme that's really essential for producing these monomers, it's probably somewhere hidden still within the genome.
So are you going to dig back in the genetic material? Is this something that you would like to look into, or you say you're happy as you are now with your 5 enzymes?
It would be interesting, definitely, from a scientific point of view, to still look into that, to see also what's capable to do with the enzyme. Especially also considering that the production of monomeric N-acetylglucosamine has also is also attractive, especially for medical applications, for regenerative medicine, for instance. Right. There are still some issues with the chemical production of these monomers. That's something we're also aiming to do in the future, potentially. But it might definitely be interesting, especially for considering such a process, to look also in more depth into this specific enzyme. Right. But currently, from the side for the production of chitosan oligomers, there's nothing like this planned.
No, because I mean, you, with all respect, there are marginal differences within these enzymes. So in a way, I mean, we're not really surprised that like mixing them all up in all different conditions actually does not really change the outcome of chitin degradation. Yeah, I would really think that it would be interesting to get as much as possible different enzymes and see what combinations of those would do with this. But there's of course lots of works and to do, and your group is still like having many years in front of them, so they can maybe look back. And of course, the main advantage, of course, of this strain is that everything can be protected still, and it's like all unknown. So this is also very important. I have like a sort of a final question. As your bacterial strain can actually also grow on this chitolyl chitin, could you see it being used, for example, to target malaria mosquitoes or something like that? Is there something that you can do with it, or do they need water to survive? Or so are there other applications than just making these chitosan dimers with it?
Highly esteemed opponent, thanks a lot for your question. There are, in fact, not maybe considering for insect as an insecticide, for instance, as you depicted. We already tested the hydrolytic supernatant from the strain on insect chitin once, but we couldn't see any substantial activity towards this. But still, there's probably some room to follow up on this as well. But there are certainly lots of different applications for the future using the strain as a whole bacterial conversion platform, for instance, as a platform organism. And for instance, that's some of the topics I addressed also in the outlook of the thesis. What we're currently looking into is utilizing the whole strain, so the hydrolytic supernatant that's capable to produce monomers and dimers for the production, as I mentioned in the question earlier, of eventually N-acetylglucosamine. To really look deeper into that, how we can optimize the process, how we can convert it into an economically interesting process, if it's even viable on a cost perspective, for instance. So lots of work to be done there, lots of development work especially. And even beyond that, last year we carried out a project in collaboration with the Chill Labs from Hosul Zoid, where we explored whether the strain could be used for the conversion of marine shrimp shells to polyhydroxyalkanolates. So these are precursors or monomers for the production of bioplastics. And we already saw that some BPO species, so that's also that's where the strain also belongs to, are capable to accumulate these polyhydroxyalkanolates. And we actually checked again the genome data that we already had available, whether there are any essential enzymes already in the genome that are responsible for the accumulation of these PHA. And there were some indications, so we carried out lots of cultivation experiments to eventually trigger the accumulation of these polyhydroxyalkanolates. But we noticed that the strain, the natural strain, is at the moment not capable to do that. But we took that as a basis, and we are now following up on that to transform the bacterium. So to convert it to a GMO by transforming it with the 3 of the most essential genes responsible for the accumulation of PHA. And that's already happened. So currently what we're looking into is whether the strain is actually capable of producing this PHA now.
Okay.
There are some considerations utilizing the growth characteristics of the strain to convert it to valorize waste material.
That sounds cool. I really appreciate your enthusiasm and also this discussion. And I'm very pleased with your answers. And I'll give the word back to the corrector.
Yes, thank you very much. The opposition will now be continued by Professor Blank. Professor Blank is a member of the assessment committee, and he's also a professor at the Applied Microbiology Institute at the RWTH Aachen.
Dear candidate, congratulations to a very nice thesis. Really going from a strain to an application. Yeah, very well done, and really not simple to do. Usually we start when the enzyme is already characterized, or doing everything in one thesis, that's really a challenge. You already touched it a little bit. What do you think about different sources of chitin? We're discussing a lot about crustacea, but there might be also, let's see, about consumer acceptance, other chitin sources coming up. And we're now, you touched already on the insects. So, and maybe there are other sources. So what are your thoughts about different possibilities? Maybe also in your outlooks, where should be the chitin coming from? Because all the crustacea are in Asia. But we are here in Europe needing the chitosan.
Highly esteemed opponent, thanks a lot for your question. Indeed, also a very interesting question to address that here, and it's also something I thought about just recently again. First of all, there are also crustaceans available in the North Sea. So we have made some contacts to some companies in Zeeland and in the Netherlands, and they're also producers for shrimp shells. And they are also eagerly looking for potential applications to convert that to something valuable at the moment. But that's just on a side note. But what you address is of course of uttermost interest at the moment, especially considering vegan products and issues with chitin from shells, from shrimp shells, posing potential allergic reactions if there's still proteins in there. So that's all something to be considered, especially for products from medical applications. And then there's also a huge issue regarding the shrimp shell biomass, that it can be dependent on seasonal changes. There can be huge fluctuations, and it's always dependent on harvests of the sea, so availability of the shrimp shells. So that's really fluctuating. And some other, of course, viable sources for chitin are currently being explored from fungal origin, of course. And it has proven to be more consistent regarding the chitin quality. It's easier to isolate that compared to shrimp shells because it doesn't require these harsh conditions that are necessary for shrimp shells. And it's also animal free. The question we have to raise here, of course, whether the strain is actually applicable to convert these sources, because enzyme specificity and selectivity towards substrate is quite essential. And we have these three allomorph versions of chitin, so the alpha chitin, beta chitin, and gamma chitin. And typically in the shrimp shells, we have alpha chitin. And it's already known that not all of the chitinases are actually susceptible to, for instance, beta chitin due to the different structure eventually. So that's a matter of substrate specificity that has to be assessed. We tested it already on insect chitin, but we couldn't see any substantial activity there of the chitin supernatant, which implies already that there is a certain specificity, or that the organism and the enzymes are already too well adapted to hydrolyzing the shrimp chitin. So maybe there are the boundaries that we look at for our strain at the moment. But that's still, it's very interesting, of course, to look for other sources for chitinases, especially to convert these substrates eventually to something, to products of interest, especially for medical, cosmetic, and food applications. So everything that has for human health context, that's very interesting.
And how about if we don't want to degrade it, just deacetylating? So I think there are applications. You mentioned it already in the medical arena for wound, and then like textiles. Which source would you aim there for? So is it still crab shells or crustacea, shrimp shells, or is it the other ones you mentioned? And you may just share your thoughts on the fungal. We are obviously using, I would like later to discuss about recombinant protein production. We use obviously fungi, but what is the vision there? How do we increase the amount of biomass that you can get the chitin from?
Highly esteemed opponent, thanks a lot for your question. That's indeed very, very difficult to address that, especially considering with enzymes. As you mentioned, using the chitin deacetylases to partially or fully deacetylate crystalline chitin, that's still one of the major issues, especially for enzymatic processes. And the reason for that lies in the fact that these chitin deacetylases mainly don't contain these carbohydrate binding domains. We saw for our chitinases that they are working very effectively on crystalline chitin, and that can be strongly associated to these carbohydrate binding domains that open up crystal structures and make the substrate accessible for the actual active site. And to actually valorize the shrimp shell material, or the chitin isolated from crustacean biomass, for instance, we would probably have to dive into enzyme evolution. And there, I would probably address creating chimeric enzymes that are so chitin deacetylases that work that can deacetylate chitin quite effectively, and then coupling it to carbohydrate binding domains to increase availability of the crystalline substrates. That would be at the moment the only viable approach to translate the enzymes into such a process. And that's also the reason, that's also explaining a little bit why we ultimately chose to go for our process to go from a depolymerization to a deacetylation process and not vice versa. Because, for instance, for the chemical processes, typically the deacetylation is carried out first to create chitosan, and then the depolymerization takes place also to increase solubility of the potential chitosan oligomers. But unfortunately for our process, we noticed that the chitin deacetylases are working quite ineffective on a crystalline chitin, and that's why we chose the depolymerization followed by the deacetylation process. But nevertheless, it would be really interesting to develop enzymes or to eventually isolate natural enzymes that would allow us to switch both processes. It could eventually allow us to produce even more versatile chitosan oligomers, and especially also oligomers with a higher degree of polymerization.
Thanks. And then maybe just a few words from your experience using E. coli as a recombinant host, and also in the context, I think it's one of your propositions, number 4. It's a really good technology to do recombinant protein production. Would you change the host if you would like to go into the direction of any commercial, or would you stick with it? And what do you see as challenges depending on the host you choose?
Highly esteemed opponent, thanks a lot for your question. There are multiple factors that actually are conditioning what host we should choose for the recombinant production. So, of course, E. coli was selected as a first strain because it's well characterized and for first study, and the production levels are decent for that. But of course, considering production yields for enzymes, there are of course strains that are known to yield way more protein yields compared to what we can achieve with E. coli. For instance, industrial Pseudomonas strains that can really go through extremely high cell densities and eventually produce have a better space-time yield. Essential for our process was a technical limitation, and that's also why we were quite happy that the secretion of the enzyme was the best working variant for all of them, because it's not required to lysate the cells, and it also cytosolic expressions, of course, can also be associated with high production levels that you have inclusion bodies eventually. But eventually, if that should be translated in a real process in the end, that has to undergo also process optimization and to really maximize yields for enzymes, because this is mostly the crucial factors that discriminated. We are comparing here enzyme production with bulk chemistry reactions at the moment for the chitosan production. We have to really make the process as efficient as possible. And that also means eventually that we have to change from E. coli strain to any high performance production strain for enzymes. Of course, it requires the folding mechanisms have to be there to oxidize disulfide bonds, and that's very essential for these organisms. But besides that, of course, everything is possible.
So, but the fungal host might be not the best idea. I'm not sure.
A fungal host.
Yeah. In industry, we use a lot of the filamentous or the yeast-like hosts because of the good secretion. But I'm not sure if it's a good idea to have a hydrolase which degrades one of the polymers of the cell wall. Yeah.
The suicide for the
fungal. Yeah, it might be a little bit odd. So I think I'm at the end, and thank you.
Thank you very much. The opposition will now be continued by Professor Bucking. Professor Bucking is professor at Fraunhofer IME and also professor at the School of Chemistry at Monash University in Melbourne.
Yeah, thank you. And thanks to be part of your PhD defense, I highly appreciate. Just to say that I'm sitting in a hotel room in beautiful Lausanne, Switzerland, participating in the EFOS conference, which is European Federation for Food Science and Technology. And so I'm very much in your topic since the last 3 days, because in this food science conference, mainly the topic is all about side stream valorization, circular bioeconomy. So we are far away from our lovely topic, aroma, food safety, and all these things. People tend to talk about plant-based proteins and how to convert something also to vegan food and things like this. So I think I'm again in the right place. What we also observe, and I think this is food science conference, that science is strongly going into the direction of commercialization. So, and being with Fraunhofer, I think it's straightforward. What I'm going to ask is all the things you mentioned in chapter 8, some outlook. And of course, you described that there is a process, and the process is quite harsh, not the, so to say, the nicest one. And as far as I understood, that was one of the triggers for you to go into some more bio-based approach. At the end of the day, people, clients, the industry will carefully evaluate the benefits. And if I talk about benefits for the industry, it's about money. Luckily, no one from industry now listens. I hope so. Our discussion will be more or less about how to convince industry. So sticking to the topic of money, can you give any rule of thumb or suggestion how you can calculate some financial outlook to your results? How you would convince industry?
Highly esteemed opponent, thanks a lot for your question. Actually, I would not just try to convince industry by going to the straight numbers, but as you mentioned, there was this one desire also to generate a green chemistry approach to deviate from the harsh chemical treatments. But the way to convince industry from the enzymatic process is lying in the potential of the enzymes to produce defined compounds. That's currently the biggest issue, especially in the chitosan and chitosan oligomer scientific community, I would call it, that lots of modes of actions and polydispersity of the mixtures produced by chemical processes are completely unknown. There are some models for that, but some chitosan oligomers are antifungal, some chitosan oligomers are antibacterial, some are anti-inflammatory, some are not. So it's really unreliable at the moment, considering the identification of modes of action and to produce reliable and consistent products. And therefore, it's of the highest interest at the moment is in producing chitosan oligomers that are defined in all 3 dimensions, what is currently not possible with chemical processes. So regarding the degree of polymerization, degree of acetylation, as well as the pattern of acetylation. And that's completely impossible doing that efficiently or at all by chemical processes. So that's the actual advantage of the enzymatic process, that we are capable to produce compounds that will never be able to produce with chemical processes. Considering it from a cost perspective, of course, that's of course quite difficult also to compare it now with the process that we have developed in the thesis, because it's not a fully developed process at all yet. We can only compare the current state of the art or the status quo of our process compared to the well-established and long-running chemical processes. But what we have to understand here is that the production costs of the enzymes, they are really making up most of the costs and of the actual process at the moment from our perspective compared to bulk chemistry with hydrochloric acid and sodium hydroxide, which is magnitudes higher, probably 1,000 times higher currently for enzymes. So in order to convert this or translate this into any economically attractive process for potential customers, we would, as discussed in the outlook of my thesis, have to maximize conversion rates by any means. So as I depicted there, by further DOE optimization experiments, by enzyme evolution, which have already shown for some chitinases that they can increase conversion rates substantially. And then lastly, of course, the recycling of the enzymes eventually to further drop the high production costs to a level where we can recycle and convert multiple batches of chitin into the certain products. It has been shown so far from these processes that are already discussed in my thesis that are still different from the process that we would have to aim to with our fully enzymatic process, that up to 11 batches of chitin could be recycled or could be converted by one batch of recycled enzymes. And I think with that, we will get into a range where it really gets interesting and we can start to compare it really with the chemical processes. But of course, as mentioned in the beginning, this always is surrounded by the fact that we can produce with this enzymatic process compounds that will never be able to produce with chemical processes.
Yeah. I mean, there's a lot of experience in cultured meat where we have the same discussion versus, I think you at Maastricht are very famous for this question. What will be the cost of a cultured meat hamburger? But in the end of the day, I think it's coming back, and you provoke, so to say. My next question is talking about the industry. Could you give some details? What kind of application? Because that's most convincing. If I say, okay, sticking to cultured meat, this has certain benefits, and of course you have some other alternatives like the plant proteins, but they of course have, we talk about highly processed stuff. We talk about extraction processes. We have to take care about some biotoxic components and all these things in cultured meat. So I think that might be a good reason for going for other options. But in your case, what would be possible? Because you said you're more specific. So the expectation would be that you could deliver certain advantage to certain markets. What would be the market, or what markets?
Highly esteemed opponent, thanks a lot for your question. That's actually something I also discussed briefly in my thesis, because the original plan for the production of enzymatic production for chitosan oligomers was to implement them in medical contexts, especially for functionalizing fibers. So to really functionalize fibers, for instance, to equip them with anti-inflammatory or antibacterial properties. For instance, for stents, for artificial implants, because there is a certain risk for inflammations or infections. And this would in general be the first market that we would aim for, because the overly advantage of these chitosan oligomers compared to chitosan is their high solubility, especially at physiological pH. And this, of course, should be exploited especially for the implementation in medical contexts, for instance. The second argument for implementing these enzymatically produced chitosan oligomers in human health context, then, of course, is again the reduction of risks and the labeling of natural product, which is still also quite considerable. And I mentioned also in the presentation earlier that there are versatile potential applications for biomedical fields, and they are also explored in lots of research topics. For instance, using it as a gene delivery system, as anti-inflammatory anti-inflammatory properties. But what's important to know is that there are barely any chitosan products on the market at the moment. Only very few products for wound healing, for instance, implementing some high purity chitosan polymers, for instance, are available. But there's nothing on the market at the moment for chitosan oligomers. And the reason for this is, again, the polydispersity of the mixtures that are created conventionally by chemical processes. So this unreliability of production for the production of consistent products is basically impeding the implementation at the moment in human health, in medicine, biomedical applications. Beyond that, of course, we should not only look at the biomedical applications, but also to come back to your background, to eventually implement it also for food applications. There have been lots of studies using chitosan as a preservative to increase shelf lives, to have edible films, to prepare functional foods using chitosan. But also there, none of the products is actually on the market readily available. And that's also probably associated, again, with the polydispersity of the products. So we're always coming back to really this issue with chitosan and chitosan oligomers, that there is really lacking a consistent production way. And that's, from my perception, the part where the enzymatic process in the end could find an actual application and to eventually also open the market for such products.
Yeah, thanks a lot. I was listening to some EFSA presentation after the European Food Safety Organization, and they take care about novel food, and novel food is not the food itself by means, but also preservatives and other things which come along and had not been in the European Union established on latest 15th of May 1997. So that's what I learned 2 days ago. So very important date. Anyway, I think that that there are some hurdles to cross in terms of regulation, and in contrast, it's a very interesting topic, which is rarely addressed as far as I can see from a food science perspective. People kill insects, extracted proteins, and make some nice insect burger and all these kind of things, looking mainly really to proteins. So this could be a very interesting contribution to circular bioeconomy. There are not too many experts on the floor. So my last question would be, you are an expert. What is your life after your PhD defense? You would like to contribute in the future, and how would this look like? What would be the first thing you would do tomorrow?
Highly esteemed opponent, thanks a lot for the question. To be honest, tomorrow already started for me last year. So already last year, I started to continue from my PhD work, working in a company. And actually, it's still associated also to some extent with the project I was working on, especially considering enzymes. And I really liked the idea to help to foster a more circular and greener environment. And that's why I'm working now in a company where we develop biopesticides to also, again, counteract any chemical analogs. And that's always also a topic of my heart now, based on what I experienced during my PhD, and that's what I'm also aiming to follow up on. I still, from a personal interest, of course, this project that I worked on now for my PhD, it always depicts that it has a really nice process already available. But from my perception, it feels incomplete because there is still so much research topics that are emerging just from this project that I would like to address. But of course, now it's not possible any longer because there's no project running on that. So that's something from a personal interest, especially to really eventually follow up on something like that.
Thanks a lot. I would like to give the word back to the prorector. Thanks a lot.
Thank you very much. The opposition will now be continued by Professor Piech. Professor Piech is a member of the assessment committee, and he's a professor at Aachen Maastricht Institute for Bio-Based Materials here at Maastricht University.
Yeah, thank you, Mr. Pareto, and yeah, dear candidate, also I congratulate you with this interesting thesis. I would like to come a little bit to the discussion of some molecular mechanisms of your material. So, and the degradation mechanism of chitosan is for sure a very exciting process. So my first question is, can you explain which chemical bonds are broken during this process in your chitin structure by enzymes?
Highly esteemed opponent, thanks a lot for your question. Considering chitinases, it hydrolyzes the glycosidic bonds between the individual monomers within the chitin structure. And to explain it in even more detail, there are chitinases available, and there are also chitosanases available. So those have different selectivities for the substrates, and eventually the chitin strand it's more or less a random distribution of N-acetylglucosamine and glucosamine. So there are actually 4 bonds that could be present within a chitin molecule. This is a deacetylated and acetylated sugar, acetylated, deacetylated, 2-acetylated, and 2-deacetylated. And what can be observed for chitinases is that those have they are more susceptible to hydrolyze the acetylated sugars. And the chitosanases are more prone for hydrolyzing deacetylated glycosidic, not glycosidic bonds, but deacetylated sugar molecules. And that's also actually something that we saw during the hydrolysis experiments by testing chitin, chitosan, and the colloidal chitin. We could see that the product spectrum that we could achieve using chitosan as a substrate is way broader compared to what we see when just using chitin as a substrate. We can achieve way higher degrees of polymerization by that. And that is due to the fact that the chitinases are not susceptible for hydrolyzing up to deacetylated sugar molecules, but they are more abundant within the chitosan molecule. So that ultimately results in a lesser distribution of the cleavable bonds, resulting then in broader product spectra. For chitin, do you also want to refer to the chitin deacetylases?
No, that's okay. I think my question was answered very precisely, and I just want to move because we have limited time. So the next question what I have is, do you know which polysaccharide is structurally very similar to chitin?
Yeah, highly esteemed opponent, thanks a lot for your question. That's of course cellulose. So cellulose is extremely similar structure-wise. And the only difference between the cellulose and the chitin in general is the presence of an amine group. And eventually for the chitin, that's also acetylated. And for the chitosan, this amine group is deacetylated.
Okay. Both polysaccharides or both materials are also quite similar because we are talking here about semi-crystalline polymers. And you mentioned also this in your presentation that this is this crystalline material or semi-crystalline material. So when I look back how the cellulases work, no? These are enzymes that can decompose cellulose. No? It is known that cellulase contains also a specific binding domain, which binds to the cellulose surface and, so to say, brings the enzyme to the right place and also plays an important role in this degradation process. So my question is, do you know if your chitinases also contain a binding domain? Are there some similarities to the cellulases in this sense?
Highly esteemed opponent, thanks a lot for your question. Yes, there are indeed so-called polysaccharide binding domains found within the proteins that was identified by the protein family database search. And and those are strongly indeed associated with binding to the chitin structure to make it available for hydrolysis of the actual active site of the enzymes. Yes, those are in there.
Mm-hmm. So I have also a question, dear candidate, to the enzymatic diacetylation of chitin. So can you summarize what are the advantages of enzymatic diacetylation compared to chemical diacetylation?
Hi, esteemed opponent. Thanks a lot for your question. There are actually multiple differences, and that's also applicable as well for the depolymerization part. The problem with the chemical deacetylation process is that it's completely unselective. So it happens absolutely random on the polymer strand, and that you cannot predict this effectively with some models by using chemicals. In comparison, the chitin deacetylases, they are already well studied, and there are multiple models developed to explain how it's possible for these chitin deacetylases to produce consistent and structurally defined deacetylation patterns. And there are already some discussions in literature how that's actually possible. So I depicted it also in my thesis that there are these multiple attack and multiple chain modes. So you have processive chitin deacetylases that just tightly bind to the chitin strand and then processively deacetylate every N-acetylglucosamine residue. Or there are these multiple attack modes where the enzyme binds to the chitin strand, then it's releasing the chitin and it's binding to another random position. But it has been found out that this is not completely random. So this so-called Subside capping model was developed to describe substrate specificity for the chitin deacetylases. So maybe to elaborate a little bit on this, there are multiple loops in the enzyme that are upstream or downstream of the actual active site on a protein level. And those are actually capable to recognize specific sequences inside the chitin or respectively chitosan strand, whether it's deacetylated or acetylated. So, and that allows the enzyme to bind to quite specific sections within the chitin strand and just deacetylates this as one specific positions. And then it can dissociate from the chitin strand and find another competitive or binding site where it binds again. So, and this ultimately allows us to predict the mode of deacetylation, and especially also, and that's the biggest advantage of the chitin deacetylases compared to the chemical deacetylation, to affect the pattern of acetylation. The pattern of acetylation has proven experimentally that it has, alongside the degree of polymerization and degree of acetylation, a substantial effect on the properties of chitosan. And therefore, it's extremely essential also, those are so-called chitosan oligomers of the third generation. So that they are basically described by those three factors. And those specific chitin deacetylases will eventually allow us to produce all potential specimen that can be produced from a chitin oligomer. There's some research being carried out, especially from a research group in Münster, from Professor Morschbacher. They are really key players inside the controlled deacetylation of chitosan. And they are actually carrying out current research to create all potential variations from chitin dimers to chitin hexamers. And those can then be produced individually. And
if I may interrupt you, I want to give a little bit of time to the final opponent, because you give a very long answer and then the final opponent out. So I would like to have the final time for the final opponent, if that's okay.
Yeah,
I'm sorry.
I'm quite satisfied with the answer. Thank you, dear candidate, for your answer. So I give in view of time, the microphone back to the prorector. Thank you.
Yes, thank you very much. That means that the final few minutes are for Professor Orru. He's also a professor at the Aachen Maastricht Institute for Bio-Based Materials.
So thank you, Rekker, for giving me the opportunity to ask perhaps one question. And I will skip, let's say, saying that you have produced a very nice thesis and go straight to my question. Actually, by listening to all the discussion so far, the most pregnant question that came up to me was, have you considered when you would scale this process to, let's say, a more industrial scale, to cascade, in fact, both enzymatic processes like the degradation using the chitosinases and the deacetylation enzymes in one process? And would that be possible? And would that then result in useful, let's say, end products?
Highly esteemed opponent, thanks a lot for your question. I'll try to answer it quickly. Of course, something like this is considered, and it's also depicted quickly at the end of the outlook of my thesis. Cascading both reactions in the same reactor, that might eventually be quite difficult, especially considering that you have to separate.
Christian Schmitz, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberations and our return. Thank you. Christian Schmitz, the degree committee here present online has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Dr. Bortesi is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the microphone.
Thank you. Do you promise to work in accordance with the principle of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible? I read your lips.
Yes, I promise I will.
By the authority vested in us by law and in conformity with the decision of the committee here present online, I hereby confer upon you, Christian Schmitz, the degree of doctor, and grant you all rights attached by custom and law. As evidence of this, you will soon receive the degree certificate signed by the rector, the secretary, and the supervisor, affixed with the official seal of the university shown by the beadle. And now I have the laudatio. Dear Dr. Schmitz, dear Christian, you didn't have what one would call an easy start in your PhD. If I say that you were a pioneer of the molecular and applied biotechnology group at IBM, it may sound like a romantic description, but it actually means that you started basically alone in an almost empty lab and had to set up everything from equipment to procedures. There was also a sudden change of plan in the topic of your research project at the very beginning, and a change of supervisor along the way. And last but not least, a continuous delay in the date of submission of your thesis due to the complicated NDA situations. So for a long time, we were not sure whether you would succeed in defending your thesis before Christmas, but here we are today, you really made it. And through this not always easy time, you managed to remain calm and focused, saw the positive side, and got the best out of its situation. It's no problem at all. It's the sentence that I heard you say most in all these years. With very little help and extra input, you quickly mastered your research topic and became the expert on T5 and chitin degradation. And even now that you've moved to work for a startup, are still part-time with us in the lab, we're very glad that we can have access to your great knowledge and expertise on this topic, which we can use to investigate new ideas and research directions that spark from your work and during your whole PhD journey. Your dedication to work and to your project is simply remarkable. More than once, I had to kindly remind you to take some holiday. And also not a single time I asked you to do something extra and last minute, which happened more often than I would like to admit. Did you say, no, I don't have time? So I very much appreciate the support and contribution also that you gave to the whole group. And this is a special moment for you, but also for our institute and for me. You are the very first to receive the doctoral degree in our research group and the first under my supervision. And as they say, you never forget your first PhD candidate. I'm very proud of you, and I'm sure you pave the way to many more doctors to come. So many congratulations, Christians, on this achievement. I wish you a lot of success in your career and all the best in your private life. Now have a well-deserved, wonderful celebration with your family and friends.
Thank you very much.
Dear Dr. Schmitz, also on behalf of the Board of Deans, I congratulate you with the honor you have acquired. And I also would like at this moment to thank the members of the jury online present for their questions and interaction. And I also thank the online audience possibly present watching this for being with us and unfortunately not coming to Maastricht, but having to do this online. I think it was a nice event. It was nice to listen to. I actually learned a lot today, so I enjoyed this session. And yeah, I think once more congratulated, and I hereby declare the ceremony to be ended.